Okay, I'm gonna go ahead and start. It's a little bit before 1:30, but I've been up since like 6 and I'm bored. And so this is going to be chapter 7, which is basically linkage and gene mapping in eukaryotes. Now, I don't think there's going to be any kind of mapping problems on the exam. For one thing, mapping in the eukaryotic organism is not my strong suit. I'm not incredibly good at it. And so if I'm not incredibly good at it, I see no reason why I should have a 200 level class do mapping. The other thing is mapping is actually incredibly hard to teach in online format. It requires practice. And basically, the only way you practice is if you sit down and work mapping problems. And eventually you get the hang of it. The other problem with mapping on exams is it takes a long time. And there's a time limit on exams. But you need to kind of know the basics of mapping in a eukaryote. Another big problem with mapping problems is that the first step is always pretty easy. But then the second step is you just have to guess. And students seem to just hate saying the second step is just a guess. And you have to pick something. And you have to work through it and see if you're right or wrong. And if you're right, well, you made the right choice. And if you're wrong, well, you have to go make another choice. And usually there's only one of two choices to make. And so you have about a 50-50 chance to make that second step go. And last couple of years, I've just decided that I'm not going to deal with that headache. Now, I think last week I finished chapter 6. And I know that I am woefully behind on posting any kind of homework assignments on Connect. And I was out of town last weekend. And so on Saturday of last weekend, I was pretty much on planes all day. Then on Sunday, I was in Arizona. And on Monday, I was basically on planes all day. And then Tuesday was a holiday, which I took off for the holiday. Wednesday, I started school again. And I had to catch up for my classes I had on Wednesday to, the days I missed. And so it took some time. But I will get assignments posted on Connect and maybe through Canvas, hopefully before the end of the weekend. Now, we're going to talk about crossover. And the really important events in crossing over occur in meiosis. Crossover can occur in mitosis, but it's much less focused upon. So we're going to define genetic linkage. We're going to mention how linkage affects the outcome of these crosses, because if genes are linked together, it goes in the face of your Mendelian predictions. Now, most animals contain a few thousand genes. And most animals, and it depends on the kind of animal, most vertebrates, for example, have about 20,000 genes in them. Non-vertebrate animals might have maybe 12,000, 15,000. However, at most, you have a couple of dozen chromosomes. Now, some species have closer to, say, 100 chromosomes or so, but most have much fewer than that. And that means that every chromosome is going to have hundreds or maybe even thousands of genes on it, depending upon the size of the chromosome and depending upon how gene dense that chromosome is. Some chromosomes are very gene dense. They carry a lot of genes in close association with each other. And some chromosomes are very, undense. And for example, one of the trisomies that is survivable is trisomy 21. And it turns out that chromosome 21 is the smallest autosome you have. It's not very gene dense. And the genes that are on it tend not to get expressed until fairly late in development. And so by that time, life is fairly well established. And so when genes are close together on the same chromosome, they are not going to assort independently. And so they will not follow the law of independent assortment. They might approximate the law of independent assortment, but they're not going to truly follow it. And so there are these things called linkage groups. And at its broadest level, each individual chromosome is a linkage group because you tend to inherit chromosomes in their entirety. And there is a fancy word called synteny. It means that the genes are physically on the same chromosome. And they tend to be linked together, which means they tend to be inherited together. And again, at its biggest level, you can describe every single chromosome as a linkage group. And so in humans, we say you have 22 autosomal linkage groups, chromosomes 1 through 22, all one, every one of those chromosomes is a linkage group. And then you say there's one X linkage group and one Y linkage group. Now, to really get an idea about the order of genes on a chromosome and how far apart these genes might be on a chromosome, you're going to have to look at least two characters in a cross, and probably more. The outcome of those crosses is going to depend on whether or not the genes are linked to each other. If, for example, Basin and Punnett discovered two traits that did not assort independently, they flew in the face of the law of independent assortment. And so they had these plants, and they had one set of plants that had purple flowers. It was homozygous for purple flowers and homozygous for long pollen. And they crossed those plants with plants that were homozygous for red flowers and homozygous for round pollen. And according to law of independent assortment, this cross should have yielded in the F2 generation a 9:3 to 3:1 phenotypic ratio. And I must admit, I changed the slide a little bit. As it is posted, this lower red rectangle includes this phenotype and the one above it. And it shouldn't. It should only occur this bottom row. Because what this says is that basically here are your parental phenotypes: purple long and red round. And in this cross, you get more than you expected of each parental type. You do get crossovers, but they're much lower than you expect based on that 9:3 to 3:1 expected ratio. And so basically what they noticed is that F2 generation had more of the parental phenotypes than it should have. At this time, it was unknown that these genes in question for flower color and pollen shape were on the same chromosome. At this time, there was no reason to expect that. Now, a crossover is an event that usually happens in meiosis, where homologous chromosomes exchange pieces of DNA. And if two genes in question are very far apart on the same chromosome, a crossover between those two genes is much more likely. If they are very close together on that chromosome, a crossover that will separate those two genes is very unlikely. And you can basically operate under the assumption if you have two genes that are on the same chromosome, they are on opposite sides of the centromere, they will essentially be inherited independently. And so the law of independent assortment will be very closely approximated. Now, working in sometime in the 1950s, I believe, there were two scientists, Creighton and McClintock. And Barbara McClintock is the one that became famous for this. But their research showed data that provided visible proof that recombinant offsprings are going to carry chromosomes resulting from that crossover. Anyway, crossing over is not a new idea by this point. One of the things that happens right early in meiosis is the homologous pair of chromosomes align and they exchange pieces of DNA with each other. They cross over. And this occurs right away in early prophase of meiosis 1. And again, it might happen in mitosis. It might happen in meiosis 2. But it yields some really interesting results when it happens in meiosis 1. And so if there is no crossing over, you say the gametes that are made by any individual are going to carry the parental combinations. They're considered non-recombinant. If there is crossing over, fancy way to say that is the non-sister chromatids of the homologous chromosomes exchange pieces of DNA. And they carry recombinant chromosomes. They're called recombinant cells. And so if there is no crossing over, the parental combination is inherited exactly. If there is crossing over, about half the gametes will carry the non-recombinant chromosomes. But the other half is going to have the recombinant chromosomes. And so you will have changed the linkage group. Now, genetic recombination can occur basically in two ways. If there is crossover in between two genes on the same chromosome, that will result in recombination. If the genes are on different chromosomes, the law of independent assortment indicates or defines that those chromosomes will be independently packaged into the gametes, and therefore each gamete will carry recombinant cells. Each gamete will be a recombinant cell. And in this chapter, the definition of recombinant offspring is defined as a gamete having a chromosome that is going to have non-parental combinations of alleles. And for example, to keep it simple, you can look at on an X-linked trait because in this case, it was fruit flies. And so by definition, crossing over cannot happen in the male fly, but it can in the female fly. And if you looked at, for example, body color, eye color, and wing length, and it turns out that all three of these genes are on the X chromosome, and you take males with gray bodies, red eyes, and long wings, which is the wild type, and you cross them with females that have a yellow body, white eyes, and miniature wings. And if the female fly is going to have a yellow body, white eyes, and miniature wings, she must inherit mutant copies of each of those alleles on both X chromosomes. And so the mutations in this case don't have a plus, and the wild type is represented by a plus. And you do that cross, you get the F1 generation. And this has all of the males better be white eyed, yellow bodied, with miniature wings. And all of the females, phenotypically, will be wild type. But genotypically, each of these will be heterozygous. And you produce the next generation. And these are the, I got to check because my email just went off. Never fails. As soon as I sit down to do a Zoom session, everybody emails me or knocks on my door. Okay. Anyway, these are the real numbers that was observed in the F2 generation. And so you have a gray body, red eyed, long wings. That's wild type for all three. And then you have the yellow body, white eyes, miniature wings, which is mutants for all three. And you have a huge number of flies in of those phenotypes. And then you have a crossover event, and you can see how this phenotype, one of these has been moved around. And you have this phenotype that crossed over. And you have a fairly large number of individuals in each of those phenotypes. And then you have this other pattern of crossovers. And you have much fewer. But, a non-zero number. And then you have this pattern of crossing over, which is basically you have none of. And this should be indicative of how far apart on the chromosomes each of these genes are. The more that you of the recombinant individuals that you see means that any of those two genes are fairly far away from each other. And if you see basically none, it means those two genes in that crossover effectively were right on top of each other. And so when you look at that data, it indicates that all three genes are on the X chromosome. It is more common that they be transmitted together as a unit. Flies with the parental phenotypes were the most common. But you did see some recombinant individuals. The parental types were the most common. But you also saw recombinants. Some were fairly common, and some were fairly rare. And so that tells you that, well, some combinations, some recombinants more commonly seen than others. Therefore, the idea is those genes that encode those traits are fairly far apart on that chromosome. The ones that had very rare recombination events were much closer together. And so if you look at the genes pairwise, you see gray body, red eyes, yellow body, white eyes. Those are parental. Vast number of them. And you look at the gray body, white eyed, yellow body, red eyed. Those are recombinants. And there's a few of those. And then you look at eye color and wing shape. Red eyes, long wings is parental. White eyed, miniature wings is parental. A lot of those. But then you have this recombination eye color and wing shape. And those recombinants are fairly common. And so mathematically, it was known at this time that chiasmata and crossing over has been observed visually in about 1909, 1910. And it was proposed this crossover, this chiasmata, involved a physical exchange of DNA between chromosomes. And T.H. Morgan kind of put two and two together. And he came up with the idea that crossing over will explain the data that he saw. And again, I have to stop because email just went off again. And I've got to go ahead junk email, but you never know when a student's going to ask you something. Anyway, in an attempt to explain what was going on, Morgan would have to draw or sketch a rough map. So here is the F1 generation being crossed to make the F2 generation. This left panel represents gametes that did not experience crossover between alleles Y and W. This panel represents gametes that were made that experienced a crossover somewhere between alleles Y and W. And there's fairly few of them, which Morgan took to mean that this allele and this allele were fairly close together. And so Morgan's big idea, likelihood of a crossover, is simply the distance between the genes that are on the same chromosome. This crossover cannot happen in the male fly because the male fly doesn't have another X chromosome. Crossover can occur in the female fly when she makes gametes because she has two X chromosomes. And the chance of crossing over occurring between any 2 points on a chromosome is just completely dependent upon how far away those two alleles are on that chromosome. And so, again, with no crossing over between anything, make a fairly large number of organisms containing the parental phenotype. And then you have these alleles, W and M, that are separated by quite a large distance. And so the numbers of recombinants is fairly high. And so it indicates that W and N are further away from each other than Y and W. And when you need to analyze that, you have to use statistics. And in this analysis, you have to use a chi-square, and it has to be able to determine whether or not your observed data match your expected data. And so you have to test that statistically. And the easiest one to do is chi-square. And so your null hypothesis is always there is no real difference between observed and expected. Your alternative hypothesis is that there is a real statistically supported difference between observed and expected. And so, from the experiment outlined previously, you propose a null hypothesis that the genes or eye color and body color obey the law of independent assortment. And from that, you can calculate the ratio of expected phenotypes. And using independent assortment, your null hypothesis is that each phenotype should occur equally. And in looking at eye color and body color, there are four possible different phenotypes. You can have the parental type or recombinant type. And so you could have the parentals or recombinations. And it turns out that in this particular cross, this F2 generation yielded 2,205 flies. That was the population size. And in this particular cross, if independent assortment was observed, gray body, red eyes, yellow body, white eyes, gray body, white eyes, etc., should all occur in a virtually 1 to 1 to 1 to 1 ratio. And you can do the math. And it turns out that if you divide 2,205 by 4, it comes out to be 551.25. And in this case, you can keep the fraction or you can throw it away. It doesn't really make a difference. And so the data is you observed 1,159, you expected 551, you observed 17, you expected 551, you observed 12, you expected 551, and then 1,017 expected 551. And it should be very easily, very early apparent. This chi-square value is going to be huge. And you have to use a chi-square table, which is back in chapter 3. You have to use you have to determine the degrees of freedom. And therefore you have two expected classes, recombinant and non-recombinant. And so n equals 2. And degrees of freedom is always n minus 1. So degrees of freedom is 1. And you look at the chi-square table, and that number of 2,100 something is much, much, much higher than it would need to be to support your null hypothesis And therefore, you reject your null hypothesis and you accept that the genes are close together on the same chromosome. They are linked. Now, Brayton and McClintock were working with corn plants, and they were looking at crossing over with a light microscope, but they could see that in maize, chromosome number 9, there were two visible peculiarities. You could actually look at it microscopically, and you could see some abnormalities in some chromosome number 9s. And this is what the chromosome number 9 normally looks like. And this is the abnormal one. On one end, it has a knob, a little swelling. And on the other end, there has been a translocation. And you can see that. You can visually see the abnormal chromosomes. And they knew that there were two genes. And they knew where they were approximately. And it was known that there was a gene for kernel color. It was near the knob. And it existed in at least two forms, colored and colorless. And there was another gene for endosperm texture that was up near here. And it existed in at least two forms, starchy endosperm and waxy endosperm. And so it was pretty well establishedly known that these things occurred kind of here. And if there was crossing over, well, it would change the arrangements of alleles that you found. And you could see that because you could see that you went from this fully abnormal chromosome with the knob and the translocation to basically a pair of chromosomes that each had one abnormality, but neither one of them had both. And so Creighton and McClintock could observe these recombination events between chromosomes. And they proposed that recombination, that result of that chiasmata, was the physical exchange of DNA from one chromosome to the other. And at the time, this was novel. When McClintock presented her findings to a panel of other, geneticists at a conference, she wrote down in her diary that these other geneticists were looking at her in awe, in horror, because until this time, it was not well established that these crossovers happened. It was not very well established that chromosomes shuffled themselves together and then broke apart when they went through meiosis. And so you don't really inherit chromosomes from your parents. You inherit a combination of chromosomes from your grandparents. And that has some very interesting connotations. And so the non recombinant chromosome number 9 had this arrangement: colorless, starchy, or colored, waxy, because that's how he set it up. The recombinant chromosomes, and you could again observe these things that recombined, are going to have, well, a different arrangements of alleles. Crossover is going to occur right through here, and you can tell that the translocation end and the knob end, instead of being on the same chromosome, went to different chromosomes. And therefore, it resulted in a recombination of the genetic material. Now, genetic mapping is useful. And again, there goes email. Okay, go back to here. Genetic mapping in plants and animals is useful for a variety of reasons. Being able to calculate the map distance between genes is useful for a variety of reasons. It allows you to construct a map showing where alleles that you, or sets of material, of genetic material, what chromosomes they are on, and how far away from each other on that chromosome they are. And that allows you some insight on how to do some genetic engineering on the organism in question. Now, genetic mapping is also known as gene mapping or chromosome mapping. And the purpose of it is to place genes in order and place genes in order as to how far away they are from each other on the chromosome. And this turns out to be a very early chromosome map of Drosophila. And this was constructed through very careful sets of crosses with alleles that caused a variety of mutant phenotypes. And this was begun back in the 1920s. It was finished sometime in the 1930s. And, this is the order of genes on the chromosome. On the left is the comparative position of the genes on the chromosome. And after molecular techniques became available, this map was reconstructed. And it turned out that the map that was constructed through just simply very carefully controlled crosses and very careful record keeping ended up to be really true. And so the molecular genetic approaches found basically the same map. The same genes were in the same position, the same distance away from each other. Now, some assumptions. Every gene has its own locus. And a locus is just a shorthand way to say a location, a physical site on the chromosome where you find that piece of DNA or that gene. Genetic maps can be used in lots of ways. One, it allows you to understand where genes are on what chromosomes. It allows you some insight on how to do gene cloning. It allows some evolutionary relationships to be inferred because organisms that are very closely related tend to have these same genes on the same chromosome. It allows an understanding of diseases because it allows you to find certain disease causing genes and know where they are. And once you know where they are, you can, find them. It allows, progresses into agriculture and breeding processes. You can make genetic maps in a couple of ways. One is recombination analysis, crossing things and getting into the F2 generation and finding recombinants and finding results of the recombinants, how many of them there are. And then nowadays, there's all kinds of molecular approaches. And it says, see in chapter 21. And I need to look, but I bet I skipped chapter 21. Because chapter 21 is entitled Genomics. And I never did genomics. So I decided to be lazy and not learn about it. So anyway, maps that are made by recombination analysis estimate relative distances between genes. And that relative distance is calculated based on the number of times a crossover occurs between those genes. And it makes what's called a linkage map. And it's very simple. Genes that are far apart have lots of recombination between them. Genes that are close together have very few. And these genetic mapping experiments typically have to be done by carrying out a test cross. And you have to look at a heterozygous individual and a homozygous recessive. Because when you see a homozygous recessive phenotype, you know the genotype of that individual. And for example, you have the P generation, long bristle gray body, wild type, short bristles, ebony body, mutant type. You do the test cross, and you potentially can get those two individuals. And then you can create the F2 generation. And you will see, relatively speaking, lots of individuals that carry the parental genotype. And relatively speaking, you will see a very few that show the crossover. And you can calculate the distance. You can calculate what they call the map distance between any two genes. And so what you're really looking at here is the number of recombinant offspring, which is taken for here, just these numbers, divided by the total number of offspring, which is adding up all of these things. And you multiply that by 100. And we say the distance between those two genes, that encode body color and bristle length, turns out to be 12.3 map units away from each other. Units of distance, and a map unit is not a physical measurement. You can't take out a very fine ruler and measure the distance between two genes. It doesn't work that way. Map units are a measure of the frequency of crossing overs. And another way to say a map unit is a centime organ. And because of that math, the S and E genes, which are bristle length and body color, are 12.3 map units across from each other on the same chromosome. And they say that each map unit is equivalent to 1% recombination. So if it's 12.3 away from each other, there should be about 12.3% recombination. Now, there is something that makes this a little bit more confusing. And there is the chances of getting multiple crossover, two crossovers between two genes, or three, or four, or whatever. If there is an even number of crossover events, two or four or whatever, DNA is exchanged. But since the crossover events occur between the genes themselves are not switched to different chromosomes. And therefore, you do not see a recombination because you inherit the genes together. And so that means you see the parental phenotypes. Therefore, the chance of multiple crossovers leads you to underestimate the actual distance between the two genes. It throws a little bit of error into your calculation. And so when you have math units that are close, you have a much more accurate measurement of detecting these things. And any given test cross is expected to only show a maximum of 50% recombinant offspring. And that is when these linked genes are going to mimic genes being on different chromosomes. And you can take one step further and you look at three factors at once. And when you look at three factors at once, it allows you to not only find the map distance between the alleles, it also allows you to calculate what order the genes are in relative to each other. And so take into account fruit flies that are going to be different in body color, eye color, and wing shape. Black body or gray body, purple or red, and vestigial or long. The pluses are the wild type, and anything indicated without the plus is a mutation. And so, step one, you have to start with true breeding lines. And so, in this case, black body, purple eyed, vestigial wings times gray body, red eyes, long wings. And there's the genotype. And that gives you the F1 generation, which you're going to have a bunch of these heterozygotes produced. And you go back and grab the true breeding line for black body, purple wings, and black body, purple eyes, and vestigial wings. And that gives you some possibilities. You could see the parental type. Let's see. A parental type. Well, there are various kinds of crossovers. Some are fairly common. Some are sort of common. And some are very rare. And the ones that are fairly common has separated wing shape from eye color and body color. And there's one there and one there. And the ones that are sort of common have crossed over in the middle. And so you have a mutant phenotype, wild type, wild type, and then wild type, mutant, mutant. And then you have the really rare ones that have had this wild type, but a mutant eye color and long wings. And so you've separated this chunk of DNA away from the other two. If those three genes have two alleles each, there are 8 possible combinations of offspring. In the case of gene linkage, the non-recombinant phenotypes should be most common. Double crossovers should be most rare. And then the other 4 groups are the result of a single crossover. And one of those crossovers is going to separate, for example, body color and separate it from wing length and eye color, and vice versa. And so basically, double crossover events are more rare. And that's because the chances of that happening twice in between two things is much more rare. And the combination of those traits in that double crossover tells you what gene is in the middle. And then to calculate the distance, you have to separate it out pairwise and start to calculate parental offspring in the F2 generation and non-parental. And that should give you number of recombinants or non-recombinants. And then the fun, you get to the math. And so they say the map distance between body color and eye color is pretty short. And you look at another pair, and they say map distance between body color and wing shape is 18 map units. And then you look at the last combination. Map distance with eye color and wing shape is 12.3. And then you can build the map. Now, at this point, in this map, you only know what gene is on the middle, is in the middle, and which ones are on either side. You do not, at this point, know which of these alleles is closest to the centromere and which one is closest to the telomere. Because at this point, either this can be the correct order or you can mirror image it. And both of those maps will satisfy the numbers that you see. In more detailed maps, then you have to put the locations of the genes according to how close they are to the centromere or the telomere. Now, note that it doesn't quite add up if you do the math. The distance between The body color wing shape should be 18.4. When you do the recombination frequencies, it gives you a different number. It doesn't take into account the slight chance that there's a double crossover in between these two alleles. You can correct for that, but I'm not going to go into any detail about that. And then there is recombination that occurs in mitosis. And recombination in mitosis happens, but it is more rare. And it's usually not noticeable. Now, mitosis doesn't involve pairing of homologous chromosomes. Therefore, crossing over in mitosis is much more rare. However, just because of the nature of how close everything is going to be in the nucleus, crossing over occasionally does occur in mitosis. And it can produce pair of recombinant chromosomes that have a new combination of alleles. And the effects of this are usually very, very small. Now, if this recombination in mitosis occurs in an early stage of development, the daughter cells that contain the recombinant chromosomes are going to continue to divide, and eventually they will result in a visible patch of tissue with characteristics that are different from the rest of the organism as a whole. And there's a guy named Kurt Stern who came up with the idea that these patches of tissue he could find on Drosophila every now and then were due to this mitotic recombination. And that became known as finding a twin spot. And so he was working with strains of flies that contained, on the X chromosome, mutation for yellow body color and wild type for gray body color, and a mutation for singed bristles and wild type for long bristles. And since this is on the X chromosome, females that are heterozygous are expected to have the gray body and the long bristles. However, when you look at it very carefully through a microscope, basically a dissecting microscope, every now and then you'll find areas that are little patches that contain the yellow body, but the long bristles, or the gray body and the short bristles. And it's spots. And so it's a little tiny, nearly microscopic, if not microscopic, phenotypic difference in regions of the fly's body. And it happens because fairly early in development, this crossover occurred. And as development continued and the fly matured to adulthood, the spot containing the crossover just got bigger. And as it got bigger, it became more obvious. Okay. That is everything for chapter 7. I don't expect to give any mapping problems, but I expect to know what linkage is and how to break linkage groups apart and some other basic things. Unless you practice mapping for a lot, it is foreign when you look at it. is I'm going to have to end there because it turns out that I cannot breathe. And so I have absolutely no idea how, well, how legible my voice is at the moment.